Yeah, good afternoon, viewers at home. My name is Bio-Edutainment Michael, the CEO of Omega Bio-Edutainment Hub, where we talk about introduction to microbial genetics, where we look at what are the basic principles of microbial genetics. And don't forget that I told you us that the basic principle for any bacterial cell or any prokaryotic organism is to the first thing is there will be duplication. For example, if this is exactly what we have, if this is a if this is a plasmid, and let's say this one is the chromosomal DNA. So what is going to what is going to take place is the first thing is going to be this DNA. This is the chromosomal DNA, and this is the plasmid. In the earlier lecture, I actually gave us the important, uh, the important, the importance of a plasmid, which I told you that it is very very special in any organism that contains it. So this is going to be replication, and through this replication, that is when we are going to have duplication of the genetic material. This is going to be as a result of binary fission. So, so it's going to be like this. The DNA, this DNA will be duplicated. The same thing is also going to be duplicated. Likewise, the information contained in it. Don't also forget that in the earlier lecture as well, I actually pointed out that there are some mechanisms that we must be used to in microbial genetics, and I told you some of the application or applicability of microbial genetics, which I mentioned about is usage in personalized medicine as well as in the, that what is the nature of the nature of the genetic material that we have in organism in microorganism, whereby we are able to look at part of the historical background of microbial genetics and so on. So under this topic today, just like I said, we are going to consider what do we mean by genetic recombination. Genetic recombination. Just from the word recombine, that is when you combine, when there is exchange of genetic material between microorganism, of genetic material between microorganism, and is actually leading to uh, new production of new set of genes, just like what we see in mitotic division, I mean meiosis division, whereby we actually look at what the mechanism of crossing over, the mechanism of crossing over in meiotic division. We will look at this in much detail as we go on. So under the genetic transfer, the genetic recombination now, we are going to look at vertical gene transfer, vertical gene transfer that we call VGT. Then the next one is a horizontal. What is a horizontal gene transfer? We will also look at it today, HGT. After that, we will come to examine the principle of a transformation, the principle and the mechanism of transformation in bacteria. We'll consider bacteria as a typical example, whereby we are going to look at what transformation actually means, and The pioneer in transformation in bacteria is Griffiths. We are going to examine Griffiths' experiments in more details. We are going to consider it in more details. After that, we'll also go. So this is going to be like under genetic transfer, mechanism of genetic transfer. This is going to be number one big topic or broad topic, followed by this, which is going to be number two, and number three now is going to be conjugation, the principle of conjugation in bacteria. So this is a new context, which is called sexual reproduction, which we can call, is not actually sexual, but is the same principle as sexual reproduction in bacteria cell. So sexual reproduction in bacteria cell, that is a conjugation in bacteria. So thereafter, we are going to look at the principle of transduction, the principle of transduction in bacteria, principle of transduction in bacteria. That is where we'll be looking at what happens in the bacteriophages. What happens in bacteriophages. Then after that, which is number 5, what are plasmids and transposons? Transposons. These are jumping genes. So what are they? Then we will now cap our today's discussion with cracking the genetic code. That is, if you want to, there's no way you will want to do anything in microbial genetics or molecular genetics without talking about the principle of genetic code. So we want to look at what are the short way of learning the genetic code by heart, so you will be able to know exactly what it means. So this is going to be the outline of what we are going to learn basically in this course. Maybe this can take us like 1 or 2 videos more. So but basically what we are going to look at is going to follow this outline. Okay, let's actually start with the first one. Let's start with the first one, which is the genetic recombination in bacteria. Don't forget the principle of central dogma theory. Whereby you have the DNA being transcribed into messenger RNA, then this will be translated, and this protein is going to be like genes in particular. So it's like the question we want to answer is, how do we have all these genetic materials being transferred from one microorganism to another? So the first one we are going to look at, just like I said, is the principle of genetic recombination. So genetic recombination is nothing but exchange. Genetic recombination is exchange of genes. Exchange of genes between segments of chromosomes, 2 segments of chromosomes, to form new set of genes. Okay, that is genetic recombination. Okay, now let's try and look at the mechanism of genetic recombination. Let's assume that this is an homologous chromosome. Let's say this is an homologous chromosome. This is okay. Let's say we have, this is A, and let's say this is B. On this chromosome, let's assume that we have we have E, F, G, H, I. Let's say these are portions on that chromosome. There are segments on that chromosome. Don't forget that homologous chromosome exists in pairs. So what that means is that this is the sister chromatid. So this is going to be small letter E, this is going to be F, this is going to be G, small g, this is going to be H, and this is going to be letter I. So during the process of genetic recombination, that is when A across each other. So let's say we now have these segments crossing to this place. This is what is called crossing over. This is called crossing over. So the sequence is the lead sequence is now going to be E, which is this, F, G, this is small, this is capital letter G. So instead of it to proceed this way, so you know this one has actually crossed to the other chromosome. So it's going to be small letter I and small letter H and small letter I. So that this is going to be a new, this is going to be A prime, then B prime now is going to be E, F, then G. So don't forget this one is crossing over to this other side. Of exchange between the genetic material on these chromosome, like E, F, G, H, I, G, H, I, and E, F, G, H, I. So this is exactly what we call genetic recombination. Genetic recombination. So recombination can take place in two different ways. It could be in form of a vertical gene transfer. Anytime you come across vertical gene with from parents or mother to the offspring. So it's a kind of vertical gene transfer is a kind of a genetic recombination whereby there is passage or exchange of genetic material between the parent bacterial cell and its offspring. That is a horizontal gene, and that is vertical gene transfer. For example, let's look at it in this way. Okay, now let's assume that this is okay, let's say for example, if there is exchange of material, this is going to be okay, this is going to be like this. It's going to be like this, so that when this is from the parents, so this is going to give birth to something like this. It's going to give rise to something like this. And like this. This one is from parents. From parents here, you can see is a look-alike like the parents. So from the parent to the offspring. So you can now see that because it is parent, that's why this one is represented in black, and this one is in blue. That's why you now see that to actually tell you that there has been transfer of genetic material from the parent to the offspring. So that is when there is that is what is known as vertical gene transfer. The second genetic transfer or genetic recombination principle is when you have horizontal gene transfer. That is a kind of transfer between two microorganisms, microorganism A and A, which has the hereditary material like this, can actually exchange genetic material with A, this is B. So if there is exchange of material between these two, which is between two different organisms, that's why this one is organism A, this one is organism B. So if there is a genetic exchange between this and this, and what is going to be something like this. After replication, you now have something like this, and we have something like this, and so that one is what is called vertical gene transfer. Okay, so that's the difference between that is difference between vertical gene transfer and horizontal gene transfer. Don't forget that I told you that anytime you come across vertical, that is transmission or transfer of genetic material from parents to their offspring, exchange of genes between parent and offspring, just as what has been. So this one is a HGT principle. Sorry, this is VGT principle, and this side is actually talking about HGT principle. Okay, so we will look at that. So immediately after genetic recombination principle, the next thing to actually consider, which is number 2, is to consider the principle or the mechanism of bacterial transformation. Okay, and that one will lead us to what I call Griffith's experiments. Griffith's experiment of between 1928 and 1944. So that was when there was still a kind of controversy on which one genetic material is it, protein or it is the DNA. So it was Griffith and his colleagues. Griffith and his colleagues like Chassis and co, that actually came out and proposed and demonstrated that the actual principle of genetic transformation will actually come from a very simple experiment which he performed and his colleagues. Now let's say there are 3 cases. There are 3 different cases. For example, now the first one is he made use of pneumococcus, and he discovered that there are 2 different types of pneumococcus. The first one is as smooth capsule, and the second one is non-capsulated. The second one is non-capsulated, which is wrinkled, non-capsulated, and it is wrinkled. So you now discover that whenever he injected encapsulated, this is the one that has capsule, so he picked it and injected it into mice. And what he discovered was that the mice died. When he injected capsulated into mice, he discovered that the mice later died. So he was now like, what could have made these mice to have died? So that was when he was now able to say that, predicted that means the difference between capsulated and not capsulated was that of capsulated must have contained some genetic material, something that will make these mice to have died. So he was able to actually show that, that means the one that contained capsule was the virulent type. He concluded that was the virulent type. Then the second thing he did was that he now got the rough but non-capsulated. So when he injected it into this mice, he also injected that into that mice, and surprisingly, the mice survived. So if you look at case 1 and case 2, you will discover that what could have made these mice to have died was that something that was virulent must have contained in the capsulated one, whereas it was absent here. So the third case was that he now hit. What he did was he killed the virulent type, that is the one that contains capsule. He killed it, and when he now injected this into the mice, he now discovered that the mice also survived. Now you should know that means there is one of the basic way to kill the virulence in this strain or this species of microorganism is to subject it into high temperature to kill it. Because ordinarily, because it is virulent, the mice supposed to have to die, but this one survived. That means the heat has actually killed that. And the third case was to, when he now added heat killed then non-virulent. It killed them and non-virulent. So that means he now injected this into the mice, and he also discovered that the mice died as well. So that is telling us that this man now believed that in the last case, under normal circumstance, the one that is already killed the mice supposed not to die. But when he combined the killed one and the non-virulent, is supposed to survive. So he was able to now predicted that this it killed virulent must have transformed the non-virulent to be virulent. That was the basic the basic mechanism or the basic prediction of Griffith, according to him. So that was when they now discover and they concluded that oh, there could be what is called transforming principle, because it was that transforming principle that transformed the heat killed, which was supposed not to be virulent again, with that one that is ordinarily non-virulent. But at the end of the day, the mice did not survive. That is the details of Griffith's experiments. Detail of Griffith's experiment. So the next thing we will consider from according to our outline, is to actually consider what is conjugation. What do we mean by conjugation in bacteria? How does it happen? Conjugation from conjugal bliss, that is the marriage between two dissimilar sexes, that is male and female. Okay, it was conjugation. Normally and ordinarily, this should not be mentioned in microorganism. However, it was later discovered, according to so many workers, that there were some types, there are some strain or some types of microorganism, especially bacteria, that they carry what is called F. pylus. F. pylus. So F plus. So they now discover that this F. pylus or F plus will behave as a male, because they will donate gene. They act as a donor. Okay, and that of F minus, and they will act as a recipient like a female. So they now discover that, like this, for example, is the case of conjugation in bacteria. So if this one is a F plus, if this is a case of a F plus, and there is a kind of coming together, and this one is a F minus, coming together of the two in order to exchange their chromosomal material, So if these two come together like this, so this place will now break up, and they will now have, so there will be exchange movements towards here. Don't forget that I said the F plus will serve as male, like the donor. So there will be movement of this genetic material towards this side, approaching towards here. So this one will donate to this. So, and it is this one. So, and F plus means that this cell is called competent cell. These are called competent cell because they have the ability, or they have the capability to be able to donate gene or exchange of gene into the one, so that this one will now become F plus as well. When you have F minus multiplied by F plus, so because this one is competent, there will be exchange of material between these, so that it's going to be F plus, or what we call HFR. So we are going to see more of this as we go on, but you should know that this kind of conjugation is very, very common in E. coli. It is very, very common in E. coli. So, in terms of recombination principle, in terms of recombination principle, you just discover that F plus has the high frequency, and that's why they call them HFR, high frequency. So we are going to see this as we actually go on. Now that one will lead us to the fourth genetic transfer principle, and that is a transduction in bacteria. Anytime you come across the word transduction, you should know transduction. So I told you earlier on that this is a genetic transfer that involves bacteriophage. This is a kind of a transfer that involves viruses and bacteria. Viruses and bacteria. So, for example, let's say this is E. coli. E. coli, and you have a phage. You have a phage. Okay. So, if this one is the DNA material, if it is injected into this, is going to now get itself integrated into the main chromosome of E. coli. It's going to get itself injected and integrated into the main E. coli, so that at the end of the day, we are going to have something like this. We are going to have something like this. So at the end of the day, so for example, now the detail of this is that whenever you have, don't forget that I told you that this is a phage DNA. The phage DNA, which is here, and this is the coat. This is the viral coat, and this is the DNA. Only the DNA or the hereditary material will actually take part in the replication, that is transferring of the genetic material. This one is going to lyse the membrane of the E. coli, and it's going to become integrated into that. So this DNA, the DNA of the virus, will become side by side. It's going to come side by side. It's going to come side by side with that of the host DNA. DNA like this, so that during the process of replication, this one is going to replicate independently. The host DNA is going to replicate independently, but it's going to become integrated. That is what is called lysogenic. So this one will become integrated into the DNA, so that into the host chromosomal DNA. And by the time this one multiply during lytic cycle, it is going to have millions of this one that is going to be produced at the end of the day. Okay. Don't forget that the fate of transduction, there are two fates. It could be, the first phase is, it could lead to what is called a lytic cycle, whereby when it gets integrated into the host DNA, this one will multiply, just like I said. And there is another one that one is going to lead into lysogenic cycle. cycle. So whereby there will not be any infection, successful infection by the host. But later on, it is going to get itself divided by divided by the use of the basic principle of binary fission. Okay, so that is exactly what is called transduction. Okay, now before we go further, the next thing to consider, the next genetic principle of a genetic transfer to consider after transduction, we will look at what do we mean by plasmid and transposon. Plasmid and transposon. Okay, now what are plasmids? Plasmid are extra chromosomal material, which can replicate independently of the host DNA. For example, when you have a bacterial cell like this, and if this is the host DNA, which is the chromosomal DNA, there are some circular material that some strain of a microorganism, bacteria, they contain. This one is capable of independent replication. So this is the plasmid. And in the earlier video, don't forget that I told you that plasmid are extrachromosomal material that are capable of independent replication. It has two fates. It can become integrated into the host DNA, and the host DNA replication will proceed. Or it can also replicate semi-autonomously. Autonomously. But what you should know is that any bacteria or any microorganism that contains this extrachromosomal material, that means they normally use it for it convert special survivability to the host. It will make them to be able to adapt very well into the host. So you cannot compare a bacteria that has plasmid with a bacteria that has only the chromosomal DNA. Majorly, it is found that those ones that lack plasmid, they are always sensitive to antibiotic. They are easily controlled by major antibiotics, whereas those ones that have plasmid, they have been found to be resistant to major antibiotic drugs. So any microorganism that contain plasmid have that survival advantage over the one that lacks plasmid. So that is what you should know. Then another thing is after plasmid, I also said transposons. There are some genetic material as well that they have the ability, they call them jumping genes. They call them jumping genes. That is, they have the ability to jump from one end of the chromosome into another end, so that they would not become integrative into them. And that is one of the major way of controlling antibiotic resistance in some microorganisms nowadays. So that is jumping genes are transposons, special genes that are called transposons. Okay, because they are jumping genes, they have the ability to jump from one end of the chromosome, or from one end of the genetic material to another end, and they become integrated, and they become also replicated along the line with the host DNA. Okay, so and these transpositions are between 704,000 base pair, are between 7,000 and 40,000 base pairs. 40,000, sorry, between 700 and 40,000 base pairs, and that one is actually very common in kanamycin resistance. Kanamycin resistance. It is found and proven that kanamycin, the gene that are resistant to kanamycin, they are transposon in nature, because they have that ability to jump from one region to another, and they become inserted. I mean, they become inverted into the host, become inverted into the host. Okay, now that is what we want to look at in the mechanism, or the mechanism of gene transfer. The mechanism of gene transfer. The next thing is, just like I told you at the early part of this lecture, that it is very possible that we should know how to crack the genetic code. We should know how to crack the genetic code. Don't forget that today we have been able to look at what genetic transfer could be, where we examine what is a genetic recombination, and that genetic recombination, that was when we mentioned crossing over, whereby there is going to be exchange of genetic material between two portions of a chromosome to give us a new set of chromosome, genetic transfer, recombination. Then under that, we look at the different types of genetic recombination, horizontal gene transfer, as well as vertical gene transfer. Then later on, we look at Griffith experiments, that is considering transformation in bacteria. Then, and later on, like that. So let's try and look at the genetic code. Let's try and look at the genetic code, cracking the genetic code. So I told you that as a student of molecular biology, most of what you should actually know is you should know genetic code very well. You should don't forget the principle of central dogma theory. Okay, then the genetic code is also another very important thing for you that you must know. Okay, now what do we do to actually know this? Don't forget, you need to remember the difference. What are the differences between RNA and DNA? The differences between RNA and DNA. Don't forget that I told you that there was a time that there was still controversy about which one is actually the hereditary material. Is it the protein, or is the DNA that is found or located in the nucleus? So the DNA contains 4 nucleotide bases: adenine, cytosine, adenine, thymine, cytosine, and guanine. Adenine, cytosine, thymine, and guanine. Whereas this in RNA, the thymine is already being replaced by the base called uracil. And apart from that, this one is a double helix structure according to Watson and Crick, and this one is just single stranded. And I told you that DNA happen to be the universal genetic material, except in some viruses whereby they have RNA as their genetic material. And the exception, okay, now let me before I go on, let me try and come back with I said central dogma theory that has to do with DNA. This one is going to undergo transcription, transcription and replication. Then we have a protein. This one will undergo the principle of translation, protein. Now, there are times that they have, like in some retroviruses where their own genetic material is RNA, not DNA. So the RNA will first of all get itself converted. That's why they call them retroviruses. So this one, instead of from RNA to be translated into protein, no. The first thing is that it is going to first of all get itself transcribed again into DNA before it's going to follow this same route. So we need to know that. Now the genetic code. Now you need to have different formulas, different formula for understanding the genetic code depending on number. But it is very, very pertinent that you at least you should have an individual way of remembering the genetic code. Now, don't forget that the genetic code, this is UCAG, because you see U, that's telling you that this one is the sequence, the base on the messenger RNA. So this is going to be like this. Okay. Okay. So this is a UCAG. Then this other side, we are just going to do it like this: UCAG, 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 UCAG. Now, don't forget what is called the triplet codon. Now, under normal circumstance, you pick this U, U, like this. Okay? But that's not exactly what we want to do today. I'll just give you very short formula. The first, this one will be 1, 2, 3, 4. 1, 2, 3, 4. On number one, if you remember, people, okay, love ice cream more than a vegetable. People love ice cream more than a vegetable. So that is telling us here. So people here is going to be sorry, ice cream more than vegetables. Okay, it is going to be okay. It's going to be 6, 3, 6, 6, 3, Okay, it's going to be 2, 6, 3, 1, 4. So when you add it together, it must be equal to 16. So let's add it now. 2 plus 6, 8 plus 3, that is 11, plus 1, that's 12, plus 4. So that is telling us that here, the first one you have P, which is for people, that is phenylalanine. That is for phenylalanine. Then love, that is you have 6. That is 2 more of this leucine. Then the whole 4 of this is going to be leucine as well. So then the one, this is going to be here, is going to be methionine, which is the starting codon. Methionine. Okay. This is going to be 3. 3 here will be isoleucine. Isoleucine. Then 1 here is going to be methionine. Then vegetable is going to be valine. VAL. So if you look at the genetic code, if you look at the genetic code, you will discover that this is what you have there. Now, talking about the second one, the mnemonics for the second one is some people talk aloud. Some people talk aloud. That is this one is just going to be 4, 4, 4, 4. So what is some people here? This first one is going to be serine, followed by people, which is proline. That's another 4. Then followed by another 4, which is theoline. Then followed by aloud, talk aloud. That is alaline. Alaline. Okay, so that is that. Now, the next one, which is column 3 of which is for alaline. Sorry, which is for adenine, is going to be TSH, okay, GALG, 1, 2, 3, 4, 5, 6, 7, GALG. Okay, LG. So if you count this, it's going to be 1, 2, 3, 4, 5, 6, 7, 8. So everything is going to be 2, 2, 2, 2, 2, like that. Okay, so that the first column here, which is 2, is going to be tyrosine. Is going to be tyrosine. Then the next 2 is going to be stop codon. It's going to be stop codon. Then followed by the next 2, which is histidine. After histidine, the next 2 is going to be glutamine. Glutamine, GAL. So then followed by the next 2, which is going to be lysine. Followed by the next 2, which is going to be asparagine, ASP. And the last one, which is going to be glutamic acid. Glutamic acid. So then the fourth column now, the fourth one is going to be 211, 4, 2, 2. If you add, this is 8. This is 4. 4 plus 4. 4, 2, 2. 2, 1, 1, 4, 2, 2. Then the last one is going to be 4. So what that is telling us is that under this, the first 2, which is this 2, is going. The first 2 is going to be 16, CYST. Then followed by 1, that is a stop codon will come in. After that, there is going to be tryptophan, which is 1, tryptophan, TRP. So the next one after that is going to be, which is going to be 4, arginine. Going to be 4, which is arginine. So after that, we have 2, which is going to be serine, SER, which is going to be arginine, ARG again, and the last one, glycine, is going to be glycine. So I think this one should be able to help you, at least to help your memory to be able to remember how to actually code for the genetic code. Don't forget that the genetic code here is going to be 5' end, 9 end. So we are going to see this, and you don't forget that these are triplets. All these are triplet codon because they are coded in 3. So for example, if you want to know what is going to code for phenylalanine, that means you combine U, which is this one, with this one, then with this one, which is UUU, then U again, for U again, UUU, then instead of this you pick C. So you pick C. So, and that is exactly how you will do and crack all the other ones to be able to understand the basic code of genetic code. Don't forget that I told you that as a student of molecular biology, genetic code is one of the things that you must know anyhow you should be able to know how you will be able to remember all this. Don't forget, I've told you that there are universal, let me say, universal code. We are going to see all this by the time we start considering